Well, thank all of you. What a wonderful treat it is to be here at this early morning hour and actually have somebody here other than just me and Corinne. So, yeah, thanks. It's really a great privilege, and I'm actually humbled to even try to take on this topic, because you notice the topic is a fairly large landscape title, Nutritional Genomics, an Overview. I mean, like, ta-da, that's like Cecil B. DeMille. Thousands of people come out and we all march to Nirvana or something. But in the 45 minutes or so that I have to share, clearly I'm going to have to kind of filter the information down to what I would hope to be some nuggets of actionable takeaways. And so there will be some glossing over, I recognize, and there will be some generalizations, and some of you may take umbrage because you'd like to dig deeper into certain things. So I will be available for discussion the rest of the day if you like, but I'm going to try to give a topographical overview as to how I see nutritional genomics, both past, present, and I'm going to even be so bold as to suggest some speculation about the future. I want to start with 1944 with Erwin Schrödinger, who you probably recognize. Sounds a little bit abstract to go to quantum physics to start a discussion about nutritional genomics, but there is a book that was put together from a series of lectures that Schrödinger gave in Dublin under the title, What is Life?, that is considered in the science of literati to be one of the most profound books ever written in the understanding of the basic substance of life. And what Schrödinger was trying to do, and being a wave mechanic guy and in quantum theory, he was trying to find a unified field theory, basically, like Einstein, for the understanding of how physics would interface with biology. And in that book, if you have the chance to read it, there are some pearls and nuggets of insight that are so applicable to today that I thought I would share a couple of them with you, because I think it sets the context for how genomics is emerging to be a tool in healthcare, because there's still a lot of uncertainty. We don't have all the answers, and probably never will have all the answers, but there are certain paths of understanding that are coming out of the work. So what did Schrödinger say in this book? And I'm taking a little license to summarize here. So he was talking about the physical aspect of the living cell, and this was not a synthetic biology discussion. This was more about how do the laws of nature relate to this complex thing that we call life. And so he then actually started talking about genetic expression. Now, I want you to recall, this is 1944. It was 1953. Watson and Crick, right? Just so I can put this in historical context. So he said that genetic expression exists in steady states similar to quantum states. And I think this is an interesting concept, because the way I interpret this is if you think of the word homeodynamics that homeostasis that we all learn about, it sounds like we have the stationary physiology around blood sugar, blood gases, blood electrolytes, and so forth, and very narrow range of control. If we get out of that, then we die. We have to control this very carefully. So it's homeostasis. But what he's really talking about is that there are these state functions that we can be in that are not in ground state, if you use the kind of model of the atom of being ground state and then excited states. And these other states are states of arousal of the organism in which it's responding to its environment, and it can be steady states, and they're homeodynamic. It's like hummingbirds' wings fluttering to keep at the flower. And so when you start looking at disease states or states of dysfunction, clearly I have a predisposition towards the word function, but dysfunction is really a manifestation of steady states of kind of quantum physiology held at an arousal state, right? So if you think, what is inflammation? Inflammation isn't a disease. It is a process related to the function of the fluttering of wings of our genetic expression patterns that hold us in a constant state of arousal at that tissue or organ level. Am I making sense here? So this is a slightly different way of thinking about the process of inflammation, because there are certain values in inflammation, certainly. There are certain values in cell replication. There are certain values in apoptosis. But when they become a steady state function, that is what we later will call a disease. So that's my first point from Schrödinger. Second was that genes regulate pore space. And this is something I never really thought about deeply, but inherent in our genes is chronology, right? It's not only depth, height, and width, but it's also time. Time is locked in by kinetics that relate to the way that stuff gets processed, and it has time. It's a clock. And I'm going to come back and talk about that how we're really working with circadian rhythms and chronobiology that are locked into our genes, tied to rhythms, right? That really connect us to our outside environment in very real ways. Magnetic rhythms, solar rhythms, lunar rhythms, tidal rhythms, all of these things are built into our genes and regulate function. So that's the second point. And then the third, and this I think really an interesting concept, is that genes are durable. And what that means is that if you've read all the books starting about the selfish gene, you probably recognize that this construct that we are the end of a tributary of a river of life that has flowed down to us, and our genes are this legacy of all these millions of years that preceded us, that this information has been processed in this long-standing experiment called natural selection that ultimately leads to us. That's a really powerful concept of the sense of ownership of the most privileged thing that we will ever be given, which is our book of life. And if we don't preserve it and we don't maintain it and we don't treasure it and do all we can to keep it from being defaced, that its durability can be impaired. It can either impair the function of subsequent generations, in the case of a replicating individual, or our own ability to function throughout the course of our life. So these are I think are really important conceptual frameworks upon which this theme of genomics is built. And that's why I wanted to set this as a context. So then I showed this photograph the other day when I presented it. It's kind of an interesting legacy for me because in this photograph, which I hope I can go back to, yes, here we go. In this photograph, which obviously Linus Pauling is the centerpiece, this was on his 82nd birthday. This was taken at Dick Humer's home in San Francisco after this big celebratory meeting that was had to honor Dr. Pauling. There are 3 Nobel Prize winners other than Dr. Pauling in this audience, and Irwin Stone is to his right, the kind of vitamin C person that Dr. Pauling picked up his thoughts. But this meeting really was focused on trying to understand what Pauling in 1949 described as molecular medicine. How do molecules of nature influence then function in such a way as it gives rise to health and disease, which was, as you know, the basis of orthomolecular medicine that was Linus Pauling's concept. And I was really struck yesterday, by the way, in that wonderful part of our program yesterday that was discussing autism. And if Dr. Pauling would have been in the audience, I think he would have been so proud and pleased. He would have stood up and said, that's what I was talking about in 1968 when I wrote the book, the article Orthomolecular Psychiatry that appeared in Science. It was a very controversial paper. He talked about mass action effects, the role of vitamins on psychiatric illness, and coenzyme effects, Michaelis-Menten constants. I mean, things that doctors don't normally think about were the fundamental basis of his thought about natural molecules influencing enzyme function that influences physiology. And so we're starting to come back to revisit that, and I'm going to be exploring that topic subsequently. So in that same year, I was privileged, I think, to write this book, which at the time I didn't actually think was that significant, but I look back now, and as I think I said the other day, it turns out to be the first time that medical and clinical nutrition were put in the title of a book, which is kind of amazing to me, actually. So medical applications of clinical nutrition, which seemed very sensible to me at the time, was actually not so sensible in the body politic. It was considered nutrition is over there, and medicine is over there, and unless you're talking about scurvy, beriberi, pellagra, thalami, and rickets, they really don't meet. And so this was the birthing, I think, I was kind of the spokesperson for the birthing of this movement, which was really starting to take shape, to take the understandings that we were starting to have about the molecular and cellular milieu and how it interrelates to disease, and start mapping what could become the 21st century medicine. So now I roll forward to the privilege of having met one of our leaders, thought leaders today in the world, Dr. Siddhartha Mukherjee. Some of you know him. He is a Pulitzer Prize winning oncologist. He's an MD, PhD immunologist. He's a wonderkind. He's truly a remarkable Renaissance individual. His book, The Emperor of All Maladies, which was about the history of oncology, that he wrote when he was at Dana-Farber, is just, it won a Pulitzer Prize. What can I say? It's a fantastic book. His more recent book, called The Gene, An Intimate History, is equally provocative and equally interested, and it's really a narrative about the whole history of the genomic revolution and its implications as he sees it both today and going forward. And I think it's It's an extraordinary, insightful book. And this review in Cell of the book was written by Eric Topol, was very complimentary. Probably you know Eric Topol down here at San Diego has written a couple of very important books, the most recent of which is The Patient Will See You Now. Just the title alone tells you the changing trend right now as people start gaining control of their own health and how genomic information relates to their own personal life in ways that is very intimate. So I think if I could give a kind of a soundbite as to where we are, and you can hold me accountable for this, you can come back later and say whether Jeff Bland was right or not. I think genomics represents the change agent that transforms all healthcare to a more precision and personalized form. And it's going to go through fits and starts. It's a complicated story, but it is the, what I would call disruptive technology that will change our perceptions of healthcare. Because there has just been this belief, certainly the belief I was trained, and I think most of you, that disease is kind of locked into our genes and it's inevitable, and we get it because we have these high penetrance of genetic characteristics that we can't avoid, and we didn't even fill out an application card, and that whole model is now really a suspect. Our genes are much more plastic and much more modifiable based on their expression patterns as a consequence of exposure to different environments. And in fact, probably hardwired genetic diseases only account for maybe 20–25% of the incidence of chronic disease in the population. So that's 75–80% then is open for discussion as to how the environment plays a role in modulating gene expression. So we're presently in the middle of this revolution in healthcare that I think is, I said unprecedented in history. That's probably a little bit of an exaggeration. I think if we would have lived at the turn of the last century, at the dawn of the age of infectious disease, we would have said that was the most unprecedented historical breakthrough in technology, because it certainly did lead to extraordinary improvements in human health and reduction of disease, understanding the concepts of infectious disease and microbes. But now we're 100 years plus downstream from that, and this is the next wave of major transition in our understanding. So significant that most of the textbooks that we're studying out of right now that teach health sciences are already out of date. They're already flawed. They're already limited in their scope relative to the new biology. And the new biology, as I said, is the agent of change that is a rule breaker, because the things that we all consider to be truths and unalienable, and we had to recite them on exams to get good grades, many of those things are now modifiable to being either wrong or not so right, one of the two. So I think that we're starting to see some really increasing acceleration of this new biology overtaking healthcare and changing our paradigm. I was very intrigued when I opened my cover of Lancet magazine earlier this year. And I have read every week of Lancet, I think, for the last 35 years, so I think I can comment on it with some degree of history. This little inside cover page was, to me, quite dramatic, because the editors and publishers of Lancet were challenging the medical community about getting on board of this revolution that's occurring in biological sciences, and that basically Lancet goes on to say, it was founded at a time of great turmoil and revolution, a period in which the Lancet, for the first time in history, made science widely accessible to medicine. And then it goes on to say, we're going to do that again. We're going to reform to make sure that we're chroniclers of the new biology that's really recreating the new medicine of today. Well, that's a really bold statement for an internationally recognized medical journal, and I think it reflects exactly the dynamics that's going on right now in the field. The rules are changing. The old guard is holding on tight to the belief of their past, but they will be replaced one funeral at a time with the new guard, and we're going to have new ideas that are going to represent really more close to the reality of biology. So let me go back to the beginning here and just give a quick historical trace. So you recall this classic quote that I pulled out that everybody probably has recited or has read. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. This is out of the short, pithy article of Watson and Crick that really changed our whole paradigm of genetics as it relates to the molecular biology. Why did I put that right next to another article, which I consider equally significant, but probably didn't get quite the play as the Watson and Crick Nature article, and that was Linus Pauling's article with his then postdoctoral student, Charles Itano, sickle cell anemia, a molecular disease. It appeared in Science in 1949. It was in the Pauling article that he talked about the point gene mutation that gives rise to the single amino acid substitution that appears in the heavy chain of globin that leads to the sickling of cells. And he was able to then characterize structure function in a really interesting way, because you know sickling is a crystallization of hemoglobin in the red cell that then packs in a different way in the cell, and then it distorts the shape of the red cell because there's so much hemoglobin in the cell as a molecule that when it packs or crystallizes and it becomes birefringent, it twists the molecule into this sickle shape, which then cuts its way through the vasculature. So this was a very powerful first example of how a single small genetic change could make such a profound change, because if you know the symptoms of sickle cell anemia, they're not in one place of the body. They're all over. Different organs are affected because it has a pan-organ effect with a single amino acid substitution, right? So it gets us thinking about, wow, small things can have big effects, and structure and function are intimately tied together, right? So you talk about molecular fits, you talk about enzymes that have specific structure that then gives rise to their function as it relates to the binding of their coenzymes or the binding of their substrates to give rise to function. So it opens up the opportunity that chemistry becomes part of our life process, which then becomes part of our understanding of health and disease. So that then begs the interesting question of whether these monogenetic diseases, which are the basis of genetic metabolism disorders, things that we study, and there are certain subspecialties in pathology that really are skilled in the art of understanding these more than 3,000 monogenetically linked homozygous recessive traits that give rise to this whole family of disorders. Certainly sickle cell is one. We have phenylketonuria, Wilson's, Gaucher's, Fabry's. I mean, the list is quite long. And so the first question you might ask is, how prevalent are these monogenetic diseases as a family of 3,000 in terms of the overall prevalence of disease in the society? And you would know that it's less than 5% of overall morbidity is tied directly to a monogenetic disease. So they're less common, but they're still, for those who have them, very significant and important. So then the question is, and this has for me been for years a really vexing question, and that is, if you have these monogenetic disorders, are they inevitable and you're unescapable, and you just are, destined to have the fulfillment of whatever the outcome is of that particular genetic disorder? And as I got to think about that, I recognized, well, no, that can't always be the case. Because if you think of some of these genetic disorders, let's use trisomy 21, or you could use phenylketonuria, you could take many examples. They exist in the phenotype in different degrees of severity. You have very high functioning people that have these disorders, and then you have people that are very low functioning. So they reflect different penetrance into the phenotype in different ways. So that suggests that the genes either have multiple alleles that reflect the conditions, or that there are other factors within the genome that regulate the expression of those mutations in such a way that they have different phenotypic presentations. So let me, if I can, use two interesting examples to describe what I'm getting to here. I'm going to use sickle cell anemia, and I'm going to use phenylketonuria, two common conditions. So the question is, can you modify the expression of these conditions by the environment, knowing that they are the extreme edge of genetic disorders, right? So if you can modify at the extreme edge, then it might say those that are less extreme might be even more susceptible to modification by the environment. Does that seem like a reasonable hypothesis? So we know that in phenylketonuria that if you restrict phenylalanine, this is well acknowledged, that you can modify the expression of the phenotype, and you can actually, the child can grow up fairly normally and have reasonable IQ, and they become less dependent on phenylalanine restriction as they grow older. They don't grow out of the genes, but they grow more tolerant to some phenylalanine in their diet as they grow older. So that's one example of changing the environment to work around a metabolic inborn error of metabolism. However, there's another part of the story, and that part of the story is going to be related back to Linus Pauling and molecular medicine and orthomolecular concepts. And that is, if you ask what it is that converts phenylalanine to tyrosine in the body, you would all know that there's an enzyme called phenylalanine hydroxylase that does that. And that's coded by the genes that are associated with the mutation that we associate then with PKU. And so you say, well, how does that enzyme actually work? It works like most enzymes do by having buddies that regulate its function. Buddies are cofactors or coenzymes. And so you might say, what is the coenzyme that then regulates or is in part associated with the regulation of activity of phenylalanine hydroxylase? And it is tetrahydrobiopterin, right? BH4. Well, tetrahydrobiopterin, remember the pterin family, which is a folic acid family. You might say, well, then if you use Linus Pauling's concepts that he described in 1968 in the Science magazine article on orthomolecular psychiatry, which is mass action, Le Chatelet's principle, if you have an equilibrium, you want to push the equilibrium a certain direction, put stress on the equilibrium. So what do? You increase the concentration of the stuff that you want to push to the other side. And so the question is, could you then give large doses of tetrahydrobiopterin to modulate the activity of a sluggish enzyme whose structure is such that its function is impaired, right? I use the baseball bat analogy here. Those of you that are baseball aficionados, I can recall when I was a kid and playing Little League and Pony League that we would always want to have the best mitts. And we wanted our gloves to be so good that if the ball even came near the pocket, it would stick in our gloves, right? So we would actually put linseed oil in our gloves, put our baseballs in there, and put it in our mattress and sleep on it. You know, this is a chiropractic delight. And so, man, you really could get, after a whole summer of sleeping on your mitt, you could really get a good pocket. Well, that's the enzyme, right? The enzyme is and the substrate is the ball. And so what you're trying to do is get such an affinity of the ball to the glove that even if you're not a good catcher, the ball's going to stick. So what happens if you have a mutated enzyme is that you didn't sleep on your mitt, right? So it's not so good a fit for the ball. Now you've got to throw the ball harder for it to stick into the glove, or you throw the ball more frequently at the glove, right? So that's my concept of Le Chatelet's principle. I'm going back to my baseball analogy. And so that's what happens in this article that's published in the New England Journal of Medicine in 2004, in which it shows that therapeutic administration of tetrahydrobiopterin to mild to moderate cases of PKU ameliorates the metabolic block, and you get stabilization of the tyrosine phenylalanine ratio. So that's kind of, to me, a support for the Pauling concept, right, of orthomolecular medicine. Now, on the left is the sickle cell anemia question, and this is a more recent review. This is 2016 in JAMA. Maybe some of you read this. It was a very nice review of fetal hemoglobin in sickle cell. And those of you who have studied sickle cell anemia or know something about its pathology know that the sickled cells that we have in adults are not found in infancy. A sickle characteristic is not expressed in infancy. And the reason for it is that fetal hemoglobin is not sickled. This may be new to some of you. I don't know if you're familiar with this. And even in the adult, we have some fetal hemoglobin. It's fairly small percentage of fetal hemoglobin. So in the sickled individual, there is fetal hemoglobin. So then the question is, could you, which is not sickled, by the way, could you then increase the concentration of fetal hemoglobin in the sickle cell individual adult so that it diluted the sickling cells below their effect to crystallize and cause the pathology? So that's the question. And the answer is yes, you can do that. And there are a number of papers. Hydroxyurea is a pharmacological compound that's used to do that. It does have some side effects. It turns out that butyrate is an epigenetic modulator. So now we start asking, gee, I wonder if the gut microbiome could have any influence on sickling in sickle cell individuals. By the way, I looked for studies on that. I could not find it, but it's an interesting question. Whether increasing butyrate by the proper microbiome metabolism could be helpful. And then the other one that's very interesting is there is an actual clinical trial. Go to clinicaltrials.gov and you'll find this on the use of sulforaphane from broccoli sprouts to modulate NRF2 to influence then the sickle cell crisis by modulating fetal hemoglobin. So what I'm trying to suggest is there are modulators in the environment which, even in the face of a hardwired monogenetic disorder, can affect the phenotypic expression of that characteristic. So this is kind of, when people think there is no hope, I think there is always hope, right? I think there is always hope. And I was 15 privileged years being on the board, scientific board of the Institute for the Achievement of Human Potential in Philadelphia, working with brain-injured children and their families, and watching miracles happen with kids that were said to their parents to be institutionalized because they were never going to be functional. And watching these kids graduate to life after I'd seen them for 10 years, starting off as just kids with cerebral palsy and serious motor neuron problems, and at 15 years of age, graduating life by doing complex gymnastics, playing violins as concert level, and then doing Shakespeare in Old English. I'm serious. I'm serious. Every year we went, my wife and I went, we're in tears. Because we recognize that miracles are only those things that we don't understand. And when you start to really understand, by opening up your mind to the possibility there's much more plasticity available than we ever fully understand, it's just finding the secret of what's unique to that individual's genome to unlock their treasure. So that's a different kind of concept in healthcare than that which we have had, which is kind of more of a deterministic model of, well, you got it and there's nothing we can do about it, so we're just going to modify the outcome by controlling the symptoms or replacing an organ or something of this nature. Okay, so this comes back to Roger Williams and Linus Pauling to me, and this is a classic photo in my photo set. This was taken in Houston in '74 at the inaugural meeting of the American Society of Preventive Medicine. As you probably know, Dr. Williams was a professor emeritus later at University of Texas, discovered pantothenic acid, past president of the American Chemical Society, and the person who really coined the term biochemical individuality. And interestingly enough, when he coined that term, he wrote an article, actually 2 articles in The Lancet. One was called Genetotrophic Disease, in which he postulates that many diseases of long latency origin are that of nutritional deprivation based on the genetic needs of the individual that takes years to express, and it becomes things like diabetes and behavior problems and basically metabolic diseases that are really diseases of overconsumptive undernutrition, too much of too little. And that was his model that he postulated. And he said there's much more variegation of nutritional need than we recognize when we're doing work on the RDAs or the RDIs, and that it's the outliers of the curve that really where you see these things. And his concept, which is a powerful concept, is everybody's an outlier for something, right? Now that's a really interesting thing if you think of our life being controlled by Gaussian curves. I call it the ghost of Gauss. You know, Friedrich Gauss visits all the time. There's 2 standard deviations from the curve of a mean of a symmetrical bell curve, when that's not even the way biology is predisposed. It's a non-symmetrical curve that most biological traits appear. And if you look at life as a topography of a variety of hundreds, actually thousands of different bell curves of everything that we are exposed to and we react to, and you run your hand over that topography, there will be something in your topography that's uniquely different out of the bell-shaped mid-cycle of everybody else. Now the question is, if it's in a very serious area, a critical area of your physiology, you call it immediate disease and you diagnose it quickly in infancy. If it's latent and it doesn't express itself for 20 or 30 or 40 years, we just call it a condition of unknown origin. You just probably had bad genes, right? So these are all linguistic ways to rationalize our lack of understanding. And we've gotten ourselves into that pattern of thinking we understand things we don't really understand. And that's why this article in the New England Journal of Medicine, The Era of Uncertainty, is so important, because the more precise we become in understanding, the more we recognize we were in an era of uncertainty, and we were just trying to make things seem certain. And then we call everything that wasn't certain an outlier, right? So this is just use of language, self-fulfilling prophecy, and getting involved in a philosophical do loop is the way I see it. And genomics is breaking us out of that model, because it's providing real information about, no, you can't rationalize that anymore. You can't even talk about comorbidities and disease adjacencies. That concept is ridiculous. It's language that really just reflects you don't understand the interconnection between osteoporosis, heart disease, and arthritis. If you understand the mechanistic connection, you can't call them disease adjacencies. You have to say they share a tributary of the same confluence of pathophysiology upstream, where you have to go to understand. So all these models that we learn, all the separation into silos that we call country clubs of ologies, they will dissolve as we start to really have true understanding and understand the origin of these conditions. Now I'm talking as a revolutionary rebel heretic here, and what I'm really reflecting is what I think is the sense of where this is going, and whether it goes that direction and is fulfilled in my lifetime, I'm not sure. But the truth will out. We are starting to understand that we're a biological system, not a collection of piece parts. that we're not separated into silos, that we've got all this integrated biology going on all the time that's interconnected to and communicating with our environment, and that interesting calculus of relationship is what determines our health and disease patterns. And that's a different model of the origin of disease than most of us learn, certainly for me. So this discovery is that the Book of Life in our 23 chapters really is communicating with our environment in very real ways in 24/7, 365. It's getting information back from all these various factors. We've got all these proprioceptor systems that are built into our body to do that, to do the sampling. Smell, sight, touch, feel, electromagnetic radiation, time zones. All these things are information concepts that our body has receptors to pick up and translate. Think of the GPCR family. These G protein coupled receptors, of which there are, I think now, over 300 that have been identified that pick up olfactants and pick up wavelengths of light and pick up electromagnetic radiation and pick up chemicals and translate it through signal transduction in cellular specific ways to alter the function of promoter regions of certain genes to regulate the expression of families of genes that then change the phenotype of the cell in response to that environment. Wow, that's really a whole different paradigm than I learned in school. So this is the major kind of transition. Genes plus lifestyle plus diet plus toxins equals our outcome. And when I wrote the book in 1999, Genetic Nutritioneering, I didn't think it was really very revolutionary, but actually now I look back and I was a little ahead of my time when I wrote this book because it introduced concepts of nutrigenomics and epigenetics and transgenerational inheritance of epigenetic factors and signal transduction and hormesis. So these were terms that were not, they were kind of used to me, but they were not really used to the body of medicine at the time. So 2002 was kind of the start of the nutritional genomics era. I've just got a little wander down history here with this British Medical Journal paper on nutritional genomics, the first paper that I could find in the medical literature that was discussing this. And then it talks about how nutrients influence genomics at all these different stages at transcription, translation, and metabolomics. And then we get into a more complex way of thinking about nutrition. And I took this, by the way, this diagram with credit from the Journal of the American Dietetics Association, which I think is very interesting, of how eating can influence the nucleosome structure in this epigenetic way to regulate how certain genes are expressed. So when we eat information that has entrance of certain library cards to go to our Book of Life and only have access to certain chapters. So it could be the chapters that relate to alarm, that we call that inflammation, or it could be the chapters of karmic bliss that lead to enlightenment and fulfillment. And so, how do we access these library cards that are going to go to selective regions of our genes that regulate their expression and function? And then we see the connection of systems biology to nutritional genomics. It's not just talking about one pathway at a time. It's really looking at a complex system. So we can't just understand body's function by looking at one gene at a time. We have to really start looking at systems of interaction. And so we learn intermediary metabolism, and we respond on exams to these pathways, in the Meyerhoff pathway, lipolysis, aminolysis, proteolysis, all of these things. We memorize them, and then we get the concept that that's how the body is working somehow. But it's not, right? This is like trying to understand a spider's web one tendril at a time, thinking, oh, I know the spider's web. I looked at that one tendril, and I really got it. But you've got to really understand the pattern of how these things are regulated, and they're regulated in time in tissue-specific ways. So what's going on in the liver is not identical, obviously, to what's going on in the GI mucosa, which is not identical to what's going on in the cardiocyte. So how are these regulated? They're regulated because there are specific marks, either sticky notes or paper clips, put on the genes of those cells. Paper clips are don't read here. They're expurgated sections of our Book of Life, and sticky notes are read here. So you hopefully have the paper clips for the heart cell regulating only the function of heart cells, right? But within that range of heart cells, you've got different functional capacities because there are metastable epithelials. Metastable epithelials are those that can be put on and taken off even as we have gone through developmental biology and embryogenesis, and they're with us throughout our whole life. So you're regulating the function of your genes by your experience of life through these metastable epithelials, right? You're putting on methyl groups, you're putting on phosphate groups, on phosphorylating certain regions of your gene, ubiquitination, acetylation. You're changing these things in real time by your activities and exposures in the planet. Now, admittedly, these are probably most vulnerable to change in infancy. However, it's now found that throughout the course of life, these metastable epithelials have transference factors, and it can be exchanged by experience. So I think we're really starting to see the origin of a precise, personalized clinical nutrition that kind of starts leveraging or accessing this information. But we're still quite a ways away from having this all nailed down. What we see is a first apparition. This is like Plato's myth of the cave. We're seeing the shadow on the wall. We're not yet actually seeing the flame of the fire. But we're starting to understand that there's a lot of gold in our hills. We just have to be very cautious and patient with how this technology develops as it gives us more illumination, and we start understanding deeper. Jose Ordovez and his colleagues at Tufts have done an extraordinary job, I think, in really helping us to understand the future of nutritional genomics, and really so much of what we call nutrigenomics, I think, has its origin back with things that they have been doing over the years. As you probably know, Ray Rodriguez at University of California, San Diego, and James Caput, who has now actually taken on the genomics responsibility at Nestle in Lausanne, Switzerland. He came from our FDA, and he came from an academic background before that. So the two of them were the authors of what is the first text on nutritional genomics, which was published in 2008. So if you think of how new this is, it's a new science. I mean, we're still on the front edge of this development. All of this stuff is happening since the Human Genome Project was announced in the Rose Garden at 2000. So this is if we think of what happens if you stopped learning at before 2000, you thought you had it nailed, you're intellectually stillborn, right? And how many people are making decisions about this based upon that they stopped really learning? And this thing that's doubling in new information every 3 years. And the first journal in nutritional genomics was in 2008. So again, it's we're really talking about fairly new technology. And then we go to where is our safe spot? Well, our safe spot is in certain areas where we have found these marker genes that really seem to have pretty significant pan-organ involvement, like SNPs within the MTHFR locus. It regulates activities associated with one-carbon metabolism. And this is where most people really start, because it's the place that we have the most deep understanding. And it gets you back into intermediary metabolism and starts to look at the complexity of cellular specific regulation of the methylome. And so we're really starting to see this, what I think is an extraordinary opportunity to delve deeper into the individual nature of how genes express themselves into function and give rise ultimately to health and disease. And with that, then, there's always for every point, there's a counterpoint. So here's my counterpoint. 2006 is this article saying, well, nutrigenomics was really overhyped, and with the blush of enthusiasm we saw in 2003 to 2005 was really not met with reality, and people overly promoted it, and they had too great of expectations, and then it led to disillusionment, and now it's falling back to the sea, and it really has no relevancy. Well, that's the pendulum overreaction, right? And there was an overhype, and there was too much enthusiasm to begin with, and people overextended what we really knew. But it's not falling back to the sea. It's just part of the iterative learning process. And as they go on to say in this article, they say currently there are steps needed to be considered in order for nutrigenomics to be applied, and that is the genes, the gene-protein network, and the strategy towards the determination of the nutrient's influence on the gene-protein expression. So it's not just one gene in and of itself by itself, it's how it regulates its protein, how that protein is regulated in its network, and how that then regulates ultimately metabolism. So it's the combination of those factors. So the American Heart Association has a position on nutrition genomics in 2016, published in Current Cardiovascular Genetics, in which they basically say it's opening up a huge opportunity for much more precise application to the individual of new therapeutics that's going to revolutionize cardiology. And of course, the PCSK9 story is really a very interesting recent example of that as it relates to what we've learned about the control of LDL and the control of atherogenesis through a specific unique SNP or a mutation that's associated with one gene. So I think that they are suggesting that the future of cardiology will be the genomic, transcriptomic, proteomic, metabolomic connection to really do a much more differential diagnosis. But like all great mysteries, riddles, puzzles, the human genome has layers of complexity, metaphoric trapdoors and false walls, and amazing hidden treasures. And those hidden treasures are things that we're still trying to find out more about. So let me give you an example of where I think a good way station to start might be. So this is a little back to the future. So some of you probably remember that one of our leaders in our field, who is a devotee of Linus Pauling, was the chairman of the University of California, Berkeley Biochemistry Department for many years, now as emeritus, and that's Bruce Ames, Dr. Ames, dearly beloved, what I would call great star of our field. One of the many articles that he's published over the years that I think should be read by everybody in this field, it's hard reading because it's very, very weighty, but I think it's really, really an important article, is this article that appeared in the American Journal of Clinical Nutrition in 2002. And the title is a little daunting for most people, this high-dose vitamin therapy stimulating variant enzymes with decreased coenzyme binding affinities, increased Michaelis constants, and how that has relevance to genetic disease and polymorphisms. That's quite a mouthful, right? But what he really talks about in this is he went through the literature with Eli Silver and Han Elson-Schwab, and they reviewed all the papers they could find that discussed the role of higher levels of nutrients on enzyme function, and in people with certain genetic SNPs, basically, different single nucleotide polymorphisms. So I don't have time to go through this in great detail, but just to summarize some of the tables in the paper, here's one of the tables that talks about potentially vitamin-responsive polymorphisms that relates to, again, the catcher's the mitt, the baseball mitt, not having a good pocket. And so you need more of the stuff in order to get the ball to stick. So he talks about methylene tetrahydrofolate reductase. He talks about NADP quinone oxoreductase, short-chain acyl-CoA dehydrogenase. We could spend time, obviously, on each one of these doing a deep drilling, so I'm just doing a very superficial overview. Glucose 6-phosphate 1-dehydrogenase, methionine synthase, foliopoly gamma-glutamyl carboxypeptidase. These are enzymes that are known to have SNPs for which they are vitamin responsive, in which the normal level of a vitamin intake for that person may be 100 or more fold the RDA, right? So when people say there's no role of nutrients in therapeutics, they really don't know this literature. They haven't studied it. And then he goes on in another table, says, what's the difference between dietary reference intakes and upper level of safety of these various nutrients? Do we have to worry about safety factors? Because often we hear people say, oh, if you're going to use high doses of vitamins, you got to worry because you can get peripheral neuropathy from B6, or there's some toxic effect. And everything is toxic at some level, even air and water. So I agree that you have to know what your safe, effective dose is. So he talks about the DRIs versus the upper limit, and you'll notice that in most cases there is 100 or more fold difference between the DRI, the dietary reference intake, and the upper limit of safety that has been studied. Well, what about cognitive function? And yesterday we heard some fantastic presentations on this whole gut-brain, gut metabolism connection to neurology. And there are many, many papers being published in this area recently, and I just chose one to kind of exemplify. This was talking about B vitamin polymorphisms and behavior, evidence of associations with neurodevelopment, depression, schizophrenia, bipolar disorder, and cognitive decline. Now, again, if Dr. Pauling was here today, he would be saying, I told you so. This was his whole model. And again, it comes back, and I'm just once again showing you the folate cycle, the one-carbon cycle that they have in this paper. It's applied in different organ systems in different ways, in different subspecialties of medicine. So the functional medicine model is, let's go to the headwaters where this and understand the headwaters, and then we don't have to go downstream and talk about the mouth of the river that's forming the sandbar in the delta, that we basically are up dealing with the action at the point of attack. And so what are the polymorphisms of B vitamin-dependent enzymes that are related to psychiatric diseases? Again, you'll notice, it's a pretty impressive list. I'm not going to go through the whole table, but he's talking about, again, these SNPs that are related to B vitamin-responsive enzymes or genes that then relate to function in the psychiatric area. So this is, again, an advancing area of understanding that has good science underpinning it, that supports this hypothesis. So what are the strengths and limitations of SNP analysis? Well, there's so many SNPs, and we're not sure which ones are really the actionable ones yet. We don't know all of many of them are probably just bystander SNPs. They don't really have much activity, but then there are some that are really pivotal in regulating function, because we know, as with sickle cell anemia, if you have an amino acid substitution in a complex protein, but it's at one critical point in that protein structure, it can have a tremendous effect on function. So that's what we really need to find out, or what are the SNPs that really regulate the more critical components of metabolism that might be upstream kind of executive function genes. My suggestion is you start with SNPs that have the strongest known clinical utility and impact upon physiological function. And this is my list of where I would want to start. We already talked about the methylation network, MTHFR, COMT, cystothionase, that family that comes out of the one-carbon pathway, which we have quite a bit of good data on at this point. Second is pharmacogenomics. And I always find this interesting. I have so many critics that I've had come up to me and say, Jeff, you talk about detoxification and the role of diet in detoxification, but that's a bunch of just speculation. There's no evidence that the diet has an effect on detoxification. And I wonder what literature they read. Because there's just literally hundreds of papers, I was going to say thousands, but I know at least hundreds, that have talked about the role of various plant constituents, secondary metabolites of plants, that regulate the expression of inducible genes to modulate the effects of cytochrome P450s and phase 2 conjugases that influences first-pass drug detoxification. There's a whole literature on that. In fact, some of you probably know that in the journal Transplant, this was an amazing article I read a number of years ago, probably 15 years ago, in which there was a recommendation that if a person went through kidney transplant or organ transplant, that the nursing staff should give them 500 cc's a day of grapefruit juice, because the grapefruit juice then lowered the expression of the cytochrome P450 that was, or the activity of the cytochrome P450 involved with the detoxification of the very expensive drug that was being given to prevent organ rejection, right? So here was a therapeutic use of a nutritional substance, grapefruit juice, to regulate detoxification. So when people say, in fact, that became, as you probably know, a drug, because they finally isolated the principal flavonoid out of the grapefruit juice that was the inhibitor, and it became a drug that's used in transplant surgery now. So I think that sometimes we just need to restate these things in different language to get over the barrier, because a person doesn't understand the connection. Next is insulin regulation, the FOXO and the FTO and fatty acid synthase gene family, the inflammatory signaling. We heard some information already previously about this, like IL-1, IL-6, TNF-alpha. Coagulation parameters, we all know about Leiden factor 5 and that relative risk of women who are on estrogenic substances for coagulation disorders. Immune factors, transglutaminase, HLA-DQA1, we already heard a lot about these as gene markers for immune dysregulation, and cardiovascular, PSK9 and CEPT and ApoB and many others. So these are where I would start with SNPs that really are more well documented to have relationship to individual response to the environment. So I will close by just saying that food is information for our genes, and that's what we've learned. And it signals in very special ways to individual tissues that are controlling how their book of life is being expressed. And I think when we put all this together, we recognize that we're at the threshold of an unbelievable transformation. We're in a transformation that to me is so powerful philosophically, because we're moving from this age of disease determinism, in which people feel, I didn't really deserve this disease, woe is me, I'm just the bad luck of the draw, to this age of plasticity and opportunistic places where we can intervene, even in the most what appear to be bleak circumstances. There may be room for modulating the way our book is expressed into our phenotype. And with that, thanks so much for your attention.